Welcome to the deep dive. Today we're tackling something huge, diabetes. I mean, the numbers are staggering. Over 537 million people affected worldwide. It's a massive burden on individuals and healthcare systems globally. And, while there are medications, lots of them actually, they don't really fix the underlying issue. The real problem is the loss of these crucial pancreatic beta cells. They're the body's insulin factories, keeping our blood sugar stable. Current treatments, they manage symptoms, but they don't replace those lost cells. Now, for type 1 diabetes, there is something, human islet cell transplantation from deceased donors. It works, it's established, but, well, the demand just massively outweighs the supply. It's a real bottleneck. But there's something really exciting on the horizon. Human pluripotent stem cells, or HPS cells, that's what we're diving into today. These cells could potentially offer a limitless supply of islet-like cells. We might be looking at a whole new era for diabetes therapy. So we'll unpack that.
Absolutely. And in this deep dive, we're going to walk you through how scientists are actually, making these beta-like cells from stem cells. It's fascinating stuff. We'll look at what the clinical trials are telling us right now, the real-world results, and importantly, we'll also tackle the big hurdles, things like the immune system, scaling up production, and the regulatory challenges. By the end, you should have a really clear picture of where this cutting-edge science stands. The core problem and the promise of stem cells.
Okay, so let's just circle back to that core problem for a second. People with diabetes, they often really struggle with keeping their blood sugar stable, even if they're incredibly careful with diet, exercise, medications. And some of the current insulin treatments, well, they carry this risk of hypoglycemia, where blood sugar drops dangerously low. That can be, really serious.
Exactly. And that's why islet transplantation, in theory, is so appealing. It's not just about pumping in insulin. It's about restoring the body's own natural ability to sense glucose and release insulin precisely when needed. It's a smart system.
A biological feedback loop.
Precisely. Something medications just can't fully replicate. But again, we hit that wall. The donor shortage for traditional transplants. It's FDA approved for tough T1D cases, sure, but the supply is just tiny compared to the need.
Which leads us perfectly to the potential game changers. Human pluripotent stem cells, HPS cells. Can you remind us what they are?
Right. So there are mainly two types we talk about. First, human embryonic stem cells, HES cells. They've been studied for longer.
Okay.
Then there are human-induced pluripotent stem cells, HIPS cells. And these are really interesting because they're made by reprogramming adult cells like, say, skin cells back into a stem cell-like state.
So you can potentially use a patient's own cells.
Potentially, yes. And critically, this approach often sidesteps some of the ethical concerns people have had about embryonic stem cells. So HIPS cells hold, well, enormous promise for regenerative medicine.
And the key thing they both share.
Their superpower, as you called it. It's their unlimited self-renewing capability. Basically, they can keep dividing and making copies of themselves almost indefinitely. This makes them a renewable and theoretically, limitless source for generating those beta cells we need.
Incredible, like a cellular fountain of youth for beta cells.
In a way, yes. And the progress just in the last 10 years or so has been, frankly, amazing. Scientists now have protocols where these HPS cell-derived beta-like cells can mature after being transplanted into the body that's in vivo. Or they can even become functional right there in the lab dish in vitro. They're showing they can secrete insulin when glucose levels rise, and structurally they're starting to look more and more like real human islets.
Okay, so the potential is clear. But how do you actually make a beta cell from a stem cell? What's the secret sauce? The recipe, the recipe. Crafting beta cells in the lab.
Right, the recipe. It's all about mimicking nature. Think about how the pancreas develops during gestation. It's a step-by-step process guided by very specific genetic switches called transcription factors and chemical signals, these signaling pathways.
So the lab work tries to copy that sequence.
Exactly. It's like trying to follow nature's own instruction manual. You can think of it like conducting an orchestra, where each signal cues the next developmental step. Or maybe like building something complex in stages, you have to lay the foundation before you put up the walls.
Okay, so walk us through those stages. What are the key steps?
Well, you start with the pluripotent HPS cells, your blank slate. The first major step is coaxing them into becoming definitive endoderm. This is an early embryonic cell type. You use specific molecules like activin A and others to nudge them in that direction.
Nudge them. I like that.
Then you guide them towards forming the primitive gut tube. From there, the really critical stage is generating pancreatic progenitors, or PPs. These are like the early builder cells specifically destined for the pancreas. This involves carefully tuning several signaling pathways, retinoic acid, BMP, FGF, SHH pathways. It's quite complex.
Sounds like fine-tuning a radio signal.
It really is. Then these PPs mature further, becoming more specialized. Eventually, you get endocrine progenitors, cells that are now committed to making hormones. And the final goal is to have these cells cluster together and mature into functional pancreatic islets, containing those crucial insulin-producing beta cells.
It sounds incredibly precise, but I remember reading that early attempts, while they might have made cells that responded to glucose, they sometimes lack something called second-phase insulin secretion. Why is that second phase important?
That's a really key point. First phase insulin secretion is that initial rapid burst when glucose levels rise. But second phase is the sustained release that follows, needed to keep blood sugar stable over a longer period, like after a meal.
Ah, so you need both for proper control.
You absolutely do. And many early protocols struggled to achieve that robust second phase. So a lot of recent research has focused on making the lab environment more like the actual body. For example, PPs naturally clump together in 3D structures. So scientists found that if they dissociated the cells and then encouraged them to reaggregate into these 3D clusters in the lab using special dishes like Agrawell plates, they could significantly boost the yield of the more mature progenitor cells. Giving them the right physical 3D environment matters.
Makes sense. What else has helped improve the recipe?
Well, specific molecules called cytoskeletal modulators have been game changers. Certain ones, like ROCK inhibitors Y27632 as an example, were found to boost the expression of a key gene, NKX61, and also help the cells survive and multiply, which is crucial for making enough cells for therapy. And other modulators, like actin polymerization inhibitors latrunculin A, for instance, can be used to very precisely trigger another important gene, NARUG3. This helps ensure you get mostly monohormonal beta-like cells, meaning they predominantly make insulin, not a mix of other pancreatic hormones.
Wow, so targeting specific genes and cell structures.
Exactly. And another big advance came from understanding the Wnt signaling pathway. It plays a role in pancreas development, but it seems you need to inhibit it at just the right time during the differentiation process. By adding a Wnt inhibitor like Wnt TC59 during a specific window, along with another molecule that boosts a different factor, ISX9, researchers found they could dramatically improve the final maturation of beta cells and basically double the number of useful insulin-producing cells they got at the end. It's like finding a hidden accelerator pedal in the process.
Okay, mini recap time. We've learned this beta cell recipe is incredibly intricate. It involves mimicking natural development stages, using 3D structures, and adding very specific molecular tweaks at precise times to push the cells towards becoming functional beta cells. That's amazing progress. But once you have these cells, how do you make sure they're truly ready, truly functional?
Good question. Because just making insulin-producing cells isn't the end goal. They need to be mature and work properly. So there are further strategies for functional maturation and enrichment. One method is reaggregation. Even after differentiation, your desired beta-like cells might be mixed with other cell types you don't want. By breaking up the clusters and letting the insulin-producing cells reform aggregates, you can effectively purify them, removing unwanted cells and significantly boosting their responsiveness to glucose, often by twofold or more.
So a purification step, essentially.
Exactly. Another key strategy is simply long-term culture. Keeping the islet-like clusters in specific nutrient media for several weeks, sometimes up to 8 weeks, allows them to mature further. This helps them develop that crucial biphasic insulin response we talked about, and it reduces the number of less mature cells.
And that longer culture time has a practical benefit too, right?
It does. It gives the final cell product a longer shelf life, which is a huge advantage for logistics, quality control, and actually getting it to patients. Think ready to use versus needing immediate transplantation.
Makes sense. Any other ways to purify them?
There's also cell sorting. Researchers identified a surface marker, CD49A, that seems to be present on beta-like cells, but not on some unwanted cell types like enterochromaffin cells. So you can use techniques like flow cytometry to physically sort and enrich for the CD49A positive cells. The challenge there, though, is scaling that sorting process up for the millions or billions of cells needed per patient. It's tricky for large-scale manufacturing.
Right, so the lab science is moving fast, refining this recipe, making the cells better and purer. But the ultimate test is, what happens in patients? What are the clinical trials showing us? 3, the clinical frontier, early results, and site selection.
Okay, let's dive into the clinical side. There are ongoing phase 1 and phase 2 trials, mostly for type 1 diabetes, but even some exploring type 2. And it's interesting because different groups are taking different approaches, which gives us valuable comparisons.
Let's start with one of the pioneers, ViaCyte. What did they try?
ViaCyte's initial approach used HES cell-derived pancreatic progenitors, remember, those earlier stage builder cells. They didn't fully differentiate them in the lab. Instead, they put these progenitor cells into special macroencapsulation devices.
Like little protective pouches?
Sort of, yeah. Designed to protect the cells from the immune system, but allow nutrients in and insulin out. They tested two main types, VC01 and VC02, implanted just under the skin.
And the results?
Well, the results were mixed. On the plus side, the transplanted progenitors did show they could differentiate further inside the body, in vivo. They started producing C-peptide in response to meals, which is a good sign C-peptide is produced alongside natural insulin.
But there's always a but.
But the C-peptide levels were quite variable between patients, and importantly, none of the patients became insulin independent, even after 2 years. A major issue they encountered was fibrosis. Scar tissue tended to form around the device implant site under the skin.
Ah, like the body walling it off.
Exactly. And that fibrosis interfered with blood vessel growth into the device, which is crucial for delivering oxygen and nutrients to the cells and getting insulin out effectively. So it hampered the long-term survival and function of the cells.
Okay, so that was ViaCyte's progenitor approach. What about Vertex Pharmaceuticals? They took a different tack, right?
They did. Vertex's strategy with their product VX880 was to transplant fully differentiated HES cell-derived islet-like cells. So cells that were already mature, insulin-producing units generated entirely in the lab before transplantation.
And how did that go? I heard some positive news recently.
Yes, the preliminary results updated in 2024 have been really quite striking. For the 12 patients who received the full intended dose, all of them achieved excellent glycemic control with HbA1c levels below 7%, and importantly, they completely eliminated severe hypoglycemic events.
That's huge. What about insulin independence?
Even better news there. 7 out of the 10 patients followed for at least 6 months actually achieved insulin independence, meaning they no longer needed external insulin injections.
Wow, that's a major leap from the ViaCyte results.
It is. And another key difference was the speed. Patients receiving the Vertex cells showed improvements in their glucose time and range much faster, often within 4 to 13 weeks, compared to the slower, more variable response seen with ViaCyte's progenitor cells.
So what's the takeaway from comparing these two?
Well, the early indication seems to be that transplanting terminally differentiated beta-like cells, cells that have already been fully cooked and quality checked in the lab, might be preferable. It seems to allow for more consistent dosing and leads to, well, better and faster clinical outcomes so far.
Okay, so what cells you use matters, but you mentioned earlier where you put them is also critical. Tell us about transplant sites.
Absolutely. Location, location, location. The subcutaneous space just under the skin is attractive because it's easily accessible, there's plenty of room, and you could potentially retrieve an encapsulated device if needed. But as we saw with ViaCyte, it's prone to that fibrosis issue, which hinders vascularization, getting good blood supply.
Right. What about the traditional site for donor islets?
That's the hepatic portal vein, which leads directly into the liver. It's minimally invasive to access and well-established. However, it has a significant downside, something called IBMR, the instant blood-mediated inflammatory response.
IBMIR. Sounds bad.
It is. When the transplanted cells come into direct contact with blood in the portal vein, it triggers a rapid and quite destructive inflammatory reaction that can wipe out a large portion of the graft almost immediately. It's a major cause of early cell loss in traditional islet transplants.
Oh, okay. So under the skin has fibrosis issues. The portal vein has IBMIR. Are there other options?
Yes, and this is where some exciting new research is happening. A site called the subanterior rectus sheath is showing real promise. This is a space within the abdominal wall muscles.
Why there?
Studies in non-human primates, rhesus macaques specifically, showed that implanting cells here promoted much better vascularization compared to the subcutaneous site and significantly improved the survival and function of the transplanted cells.
Fascinating. Has this been tried in humans?
Yes. There was a really notable clinical trial reported by Wang and colleagues in China. A patient with type 1 diabetes received autologous HIPES cell-derived islets, meaning cells derived from their own body transplanted into this subanterior rectus sheath site.
Autologous, so no immune rejection issues ideally. What happened?
The results were remarkable. They detected C-peptide production just 2 weeks after the transplant. And incredibly, the patient achieved insulin independence after only 3 months.
3 months? That's incredibly fast.
It really is. Another advantage of the site was that they could monitor the graft non-invasively using standard ultrasound and MRI, which is important for checking cell survival and ruling out any potential tumor formation, like teratomas.
Wow. Any other promising trials like that?
There was another report from China by Wu and colleagues involving a patient with type 2 diabetes. This patient received autologous HIPS cell-derived islets via infusion into the portal vein and also achieved insulin independence, which was maintained for 56 weeks at the time of reporting.
So success in both type 1 and type 2 using autologous cells.
Yes, although it's important to note, particularly for the type 2 patient trial using the portal vein, that patient was already on immunosuppression medication for a prior kidney transplant. So while the results are fantastic, we still need more data from larger groups of patients, followed for longer periods, especially regarding the long-term function and safety, and particularly the immune response in patients not already immunosuppressed.
Okay, another mini recap. We're seeing real clinical progress, especially with fully differentiated cells like Vertex's VX-880 showing insulin independence. And new transplant sites, like the subinterior rectus sheath, look very promising, especially with autologous cells derived from the patient themselves. But big challenges remain. The immune system is still a major hurdle for most patients, and then there's the sheer scale needed.
Exactly. Those are the next big mountains to climb. How do we stop the body from rejecting these cells without harsh drugs? And how do we make enough of these cells for potentially millions of people?
Well,
5E, overcoming obstacles, immunity, scale, and regulation.
right, let's tackle the immune system first. That's kind of the elephant in the room, isn't it? Most of these trials, except those autologous ones in China, use allogeneic cells from a different donor. And that means
That means lifelong immunosuppression drugs for the patient. These drugs prevent the body from rejecting the transplanted cells, but they come with their own set of risks and side effects, increased susceptibility to infections, potential kidney damage, even increased cancer risk. It's a significant burden.
So finding ways around immunosuppression is critical if this therapy is going to be widely applicable. What are the main strategies being pursued?
There are several exciting avenues. We already mentioned autologous cells using the patient's own reprogrammed cells. The Chinese trials show the potential, but as we noted, the immune tolerance aspect needs more study in patients not already on immunosuppressants.
Okay, what else?
HLA-matched cells. HLA, or human leukocyte antigen. These are proteins on our cells that the immune system uses to recognize self versus non-self. The idea is to create HPS cell-derived beta cells that are a close HLA match to the recipient, minimizing the chance of rejection.
Like matching organ donors.
Exactly like that. Progress is being made. You can generate HLA-matched HIPSA cell lines relatively quickly now, but the regulatory process to qualify each new line for clinical use is, well, long and very expensive.
So not practical to make a custom line for every single patient.
Probably not for widespread use. That's why the concept of an HLA haplobank is gaining traction. The idea is to create a bank of maybe 10 to 100 different HIPSA cell lines, carefully selected from donors who are HLA homozygous, meaning they have 2 identical copies of key HLA genes. Calculations suggest a bank of this size could provide a reasonably good HLA match for about half the population in diverse regions like China, Japan, the UK, and the US. It's a more feasible approach to providing off-the-shelf matched cells.
That's clever. But for type 1 diabetes, isn't there still the problem of autoimmunity, the original disease process that destroyed the patient's own beta cells?
That is a huge concern. Even if the transplanted cells are HLA matched, the patient's immune system might still recognize them as beta cells and attack them, just like it did their original ones. Researchers are trying to figure out if matching specific HLA types or avoiding certain diabetogenic HLA variants could help reduce this autoimmune recurrence. But honestly, the data is still pretty limited and complex.
So matching might not be enough on its own for T1D. What about hiding the cells?
Right, that leads to immune-isolating encapsulation devices. We talked about ViSite's macro devices. The goal is a physical barrier permeable to nutrients and insulin, but impermeable to immune cells.
But ViSite had issues with fibrosis.
They did. Vertex also tested an encapsulated device approach with their product VX264. The first patient was implanted without immunosuppression in 2023. However, unfortunately, more recent updates in 2025 indicated that the device wasn't allowing for sufficient insulin production, and that trial has been halted. So encapsulation is proving trickier than hoped.
Okay, so if matching is complex and encapsulation is challenging, what's left?
This is where the really cutting-edge stuff comes in. Hypoimmunogenic cell lines, making stealth cells. The idea here is to use genetic engineering, often with tools like CRISPR, to modify the HPS cells themselves so they become invisible to the immune system.
How do you make a cell invisible?
Well, one approach, pioneered by companies like Sana Biotechnology, involves knocking out the genes responsible for HLA class 1 and class 2 proteins, basically removing the flags the immune system looks for. At the same time, they might overexpress a protein, like CD47, which acts as "don't eat me" signal to certain immune cells.
Wow, stealth mode activated.
Pretty much. Studies in animal models, including humanized mice and non-human primates, showed that human islets engineered this way could avoid both rejection and autoimmunity and successfully treat diabetes. Sana is planning phase 12 trials. CRISPR Therapeutics is also running a trial with their own hypoimmunogenic cell line, which has multiple gene edits. This is a really hot area of research.
That sounds incredibly promising for avoiding immunosuppression. Okay, let's shift gears. Assume we solve the immune problem, either with stealth cells or matching or something else. How on earth do we make enough cells for potentially millions of patients? The scale seems daunting.
It is daunting. Scalability is a massive hurdle. Think about traditional islet transplants. They typically require over 7,000 islet equivalents, or IEQs, per kilogram of body weight. For an average adult, that's maybe 450,000 IEQs, often needing islets from more than one donor pancreas.
And stem cell-derived ones might need even more.
Current trials suggest yes. Doses are often in the range of 1 to 1.4 million IEQs per patient. Producing that many functional islet clusters is a huge manufacturing challenge. Traditional lab culture and flat dishes just won't cut it.
So what's the solution?
The field is moving towards 3D suspension bioreactors. These are essentially large tanks where cells can be grown suspended in nutrient liquid, often attached to tiny microcarriers, or encouraged to self-aggregate into clumps. This allows for much higher density growth and vastly greater cell numbers compared to planar cultures. You can expand the HPS cells maybe 20 to 50-fold with each passage or round of growth in these bioreactors. There are different types: roller bottles, spinner flasks, stirred tanks, vertical wheel reactors, all aiming for large-scale, controlled expansion and differentiation.
But even one bioreactor might not be enough for one patient dose.
Probably not. A moderately sized bioreactor, say 0.3 liters, might yield somewhere in the range of 10,000 to 100,000 IEQs. So yeah, each patient's dose will likely need to be pooled from the output of multiple bioreactors run in parallel. It's a complex orchestration.
Okay, so you can potentially make enough cells. But for a truly off-the-shelf therapy, you need to be able to store them, right? Like freezing them.
Exactly. And storage is another significant challenge. Traditional cryopreservation freezing cells slowly often involves breaking the islet clumps down into single cells, which unfortunately kills a lot of them during the freeze-thaw process. Even trying to freeze the 3D clumps directly often results in poor viability, and the cells might need days to recover afterwards, which isn't ideal.
So freezing is tough. Is there an alternative?
There's a newer technique called vitrification. This involves ultra-rapid freezing, essentially turning the water in and around the cells into a glass-like state without forming damaging ice crystals.
Like snap freezing.
Sort of, yeah. Lab studies have shown that HPS cell-derived islet clumps vitrified for months can maintain really high viability, over 90%, and still function perfectly well to reverse diabetes in mice after thawing.
That sounds much better. What's the catch?
The catch is scaling it up and keeping it sterile. Current vitrification methods often involve handling relatively small amounts of tissue, maybe on a small mesh, and might involve directly plunging samples into liquid nitrogen, which raises sterility concerns for clinical products. So, while vitrification is promising, making it work reliably and sterilely for the vast number of cells needed per patient dose is still a major engineering hurdle.
So for now.
For now, on-demand manufacturing, growing the cells in bioreactors specifically for upcoming transplant needs, seems more practical than relying on large banks of frozen or vitrified cells, though improving storage is definitely a high priority.
Okay, so we have the science, the clinical trials, the challenges of immunity and scale. But how does all this cutting-edge stuff actually get approved and delivered safely and consistently to patients? Let's talk regulation.
Regulation is absolutely crucial. You have these incredibly complex living cell therapies. Ensuring they are safe, effective, and manufactured to a consistent high quality is paramount. Regulatory agencies worldwide, like the FDA in the US, EMA in Europe, and others, are working closely with developers to establish frameworks. It all falls under the umbrella of good manufacturing practices, or GMP, and robust quality management systems. These are governed by specific laws in each region, but also guided by international standards from organizations like PICS, ICH, and the WHO.
What are some of the key things regulators focus on for these therapies?
It starts right at the beginning with the testing and qualification of starting materials. That means rigorous screening of the original cell donors for any infectious diseases, hepatitis, HIV, Zika, West Nile, you name it, depending on geographic risks. The HPS cell lines themselves need extensive testing to confirm their identity, ensure they are genetically stable over time, and check their basic potential, their potency. Master cell banks and working cell banks are created under strict controls.
What about other stuff used in the process, like the media the cells grow in?
Good point. Those are called ancillary materials. Things like enzymes, growth factors, culture media components used during manufacturing but not intended to be part of the final product. These also need careful qualification based on their potential risk. Materials derived from animals or humans carry the highest risk and require the most stringent testing and documentation.
Okay, so starting materials are checked. What about the manufacturing process itself?
GMP for manufacturing is incredibly strict for cell therapies. The entire process, from thawing the initial cell vial to final formulation, has to happen in highly controlled cleanroom environments, typically meeting ISO 5 or Grade A air quality standards, often using specialized isolator cabinets to minimize contamination risk.
Is that stricter than how traditional donor islets are handled?
Yes, generally much stricter. This has actually led to some regulatory discussions, as the requirements for these manufactured cell products are often more rigorous than for minimally manipulated donor islets. Workshops involving the WHO and FDA are trying to harmonize approaches. And a key trend here is automation. To achieve industrial scale and ensure consistency, manufacturers are looking to automate as much of the process as possible using systems similar to those developed for CAR T cell therapies. Automation removes human variability and subjectivity, which is critical for producing a reliable product batch after batch.
Makes sense. So you've made the cells under GMP. What checks happen before they can actually be given to a patient?
That's the final hurdle: quality control, QC, and release criteria. A whole battery of tests is performed, both during the manufacturing process, in-process controls, and on the final cell product before it's released for clinical use. These define the critical quality attributes, or CQAs, of the product.
What kinds of tests? Safety must be top of the list.
Absolutely. Safety testing includes checking for bacterial contamination, pyrogens, and endotoxins, basically fever-inducing substances. A crucial safety test for HPS cell products is ensuring there are no residual undifferentiated stem cells left. Even a tiny number, perhaps as few as 100 cells in some models, could potentially form teratomas, benign but unwanted tumors containing various tissue types.
How do they detect such small numbers?
Using highly sensitive methods like digital droplet PCR or specialized flow cytometry assays designed to pick up markers specific to undifferentiated HPS cells. It's a critical release test.
Okay, safety first. What else?
Identity. Confirming the cells are indeed the pancreatic beta-like cells they're supposed to be, using specific markers or genetic tests. Purity. Ensuring minimal contamination from other cell types generated during differentiation or from residual ancillary materials. Dose. Accurately measuring the number of viable cells, often correlated with those IEQs we discussed, perhaps using flow cytometry to count the proportion of actual beta cells. And then, arguably the most complex one, potency. This isn't just about counting cells, it's about proving they work. Do they express the key transcription factors needed for beta cell function, like PDX1, NKX1, MAFA? And most importantly, can they perform a glucose-stimulated insulin secretion assay, or GSIS? This lab test confirms they release insulin appropriately in response to different glucose levels, ideally showing that biphasic pattern we talked about, mimicking natural islets. This is the ultimate proof of function before transplantation. Finally, stability testing ensures the product maintains its quality over its intended shelf life.
Wow. That's an incredibly thorough process, from starting materials right through to the final functional checks. It really highlights the complexity of bringing these therapies to patients safely and effectively.
It really does. It's a massive undertaking requiring expertise across biology, engineering, manufacturing, and regulation.
Okay, let's try and bring all these threads together now. What's the big picture for the future? Outro. So we've journeyed through this really, truly exciting landscape of stem cell therapy for diabetes. From the meticulous science happening in the lab, crafting these beta cells, to the promising but also challenging results we're seeing in early clinical trials, and finally navigating that complex web of immunity, scale, and regulation.
And the potential, despite the hurdles, remains immense. The prospect of having a potentially limitless source of functional glucose-sensing beta cells for patients who need insulin, it could fundamentally change how we treat diabetes. It could dramatically improve quality of life for millions.
So looking ahead, what are the absolute top priorities for the field to make this a reality?
Based on where things stand, I'd say there are a few key areas. First, continuing to optimize the differentiation protocols to get beta cells that function even more like natural islets, ideally reducing the number of cells needed per patient. That helps with scalability too. Second, cracking the code on robust cryopreservation or sterile vitrification. That's essential for having a truly practical, off-the-shelf therapy that can be shipped and stored easily. Third, and this is a big one, developing those hypoimmunogenic stealth cells, or reliably effective HLA matching strategies or immunoprotective devices to finally eliminate the need for chronic immunosuppression. That would open this therapy up to far more patients.
And the fourth.
Fourth, standardizing global regulatory guidelines, ensuring there's a clear, consistent, and efficient pathway worldwide to approve these therapies and ensure equitable access and consistent product quality no matter where a patient lives.
It sounds like a massive collaborative effort is needed.
Absolutely. This isn't something one lab or one company can solve alone. It requires deep, ongoing collaboration between scientists, clinicians, engineers, regulatory agencies, industry partners, and, very importantly, patient advocacy groups. Everyone needs to be pulling in the same direction.
It's truly inspiring stuff. So let's leave our listeners with this thought. Imagine a future where being diagnosed with diabetes doesn't automatically mean a lifetime of injections and finger pricks, but perhaps a single treatment, a curative cell therapy. As this science continues to race forward, what ethical or societal questions do you think we need to grapple with most urgently to prepare for that future? Something to ponder. Thank you for joining us on this deep dive into the future of diabetes treatment. We hope you found it as fascinating as we did. Stay curious, stay informed,